So hopefully that movie was a fun way to kind of educational and fun to see how different hairs are pretty much produced and what we do could change that. So here we have a hair follicle. So here's the gland. This is where the hair is growing and it comes out through here. Well, depending on your ethnicity or the type of hair you have, it determines whether you have straight, wavy, or curly, and it's kind of based on pretty much your gland, how it's shaped, and then also the circumference or the cross section of your hair right here. So thin ones are a little bit will give you more curly or wavy hair, while round, thick will kind of give you straight hair. Here's kind of just a fun pic of what's going on. Here you can see different types of animals. So you can see like dogs and cats are very small, deer, rabbits are a little bit thicker. But here's human hair. And what I want you to notice is there's the cuticle on the outside. That's what protects it. Also, that kind of gives it the shine or the damage depending on how you're going to see it. So that's why shampoos will have stuff to try to condition this, moisten this, or make it shiny. And then you have a center or a core, and then you have the cortex on the outside. And that's kind of what we want to focus on, is this outer part right here. This is where the proteins are, or keratin, and this is kind of the property of hair, and this is where those chemicals like lye or sodium hydroxide is trying to attack to kind of change your hairstyle. So you could do things like put water on it. Those are all temporary. Water will kind of break hydrogen bonds and stuff, or help relax those bonds. But then once it kind of goes away, it's going to naturally go back to what it's supposed to be. You could also use a heat iron, which will kind of temporarily push it into a certain order, but as it relaxes or you shower, it's going to go back to normal. So to permanently relax it, you need to have a chemical reaction happening. In order to do that, you're breaking certain things. So here's some couple things that you might see. So here's the hair. Here are the keratins and the proteins that are going to be attached to each other. So sometimes you have strong bonds in between the protein itself and other neighboring proteins. Sometimes you have weak ones. So here's a really strong bond. So this right here, this is covalent. I guess what? Covalent. So that means it is a very, very strong bond. Here you have ionic, and then here you have hydrogen. So this is the weakest, next strongest, strongest. So when we're talking about relaxers, they're going after this right here and breaking it. Other methods, you could kind of temporarily break the hydrogen bond, but it does come back together. Even ions, if you add water, will help break that bond, but it's going to come back together eventually. But this, you can't break it with water or heat. You really need a chemical, in this case a relaxer, and that's what we watched last week. Now here's kind of example of the opposite. So this is kind of to curl your hair, which is the same kind of idea. It's a little bit different because you want to reform those bonds, where a relaxer you just want to get rid of them. So here you can notice that the sulfide bonds are nice and straight. You want to break it with a relaxer. This is a non-lye one. This is just a salt, kind of a salt that you go in, and it breaks like the sulfide bond. Then you can kind of curl it, and then you use something like hydrogen peroxide to kind of reform those sulfide bonds again. So when you go get your hair permed, that's what you're doing. You are putting some salt in to break the bonds, putting in curls, using hydrogen peroxide to kind of push them all back. Now for straighteners or relaxers, the opposite direction, and hydrogen peroxide goes this way, but you don't form those bonds anymore. It's just gone. So it pretty much does this, and you can't undo it. That's also why when you straighten your hair using relaxers, it damages it. So you're kind of stripping away not only the nutrients on the outside of the hair, you're also changing inside, so they become brittle. So if you dye your hair a lot, or you bleach your hair a lot, or even straighten your hair gets really brittle. So it's easy to get like splitted and stuff, because you are kind of physically changing your hair and damaging it, which you can't undo. Here's another example. This is more of a protein itself, but you have these disulfide bonds right here, and you break it apart, which relaxes the protein or amino acid, and then it can have a different shape. Okay, so these are really cool because what I like about it kind of gives a really quick introduction to kind of the different kind of interactions that proteins can have or amino acids can have with each other. So with hair, it's mostly disulfide bonds. When we look at that chart that I believe we had the other day, here are all the different properties of it. And I asked you, okay, so you have the non-polar ones, you have the acid ones, you have the base ones, you have the polar ones right here. Some are aromatic and some are hydroxyl groups, but the main thing is these have oxygen, and you have others. So cysteine is one of those others because it has, instead of oxygen and hydrogen, it has sulfur and hydrogen. And it could also be polar, but it also can create bonds with each other. So that's kind of what made it special. It can make disulfide bonds. Today we're going to focus more on these two right here. These don't make bonds, but they make ions. And ions can make bonds with other ions itself. So here are some amines. Let me get rid of this. So you'll notice that you have nitrogen and hydrogen. So these are going to be able to share the hydrogen a little bit differently, and they're going to willingly take some hydrogen. So if you look at our next slide, notice that it takes up hydrogen, so they become positive. You'll also know right here, here's the carboxyl group, and then here's the amine group of our protein, and this is technically the R group. I'm going to kind of ignore these two. And the reason why I'm going to ignore it, because when our amino acids is in the protein structure, these are in bonds. Okay, so this is attached to another amine, and this is attached to a carboxyl group. So they're going to be in a bond, so we really don't care whether or not by itself it's going to be positive or negative. We really only care about the R group. So for this one, we care about what's happening to the nitrogen. Here we have some acids. And the exact opposite. These were proton acceptors. They're willing to take up hydrogen. These are going to be proton donators. So when you put them in certain pHs, it's going to give up its electrons, or sorry, its protons. Now this chart is really cool because it shows you, like I said, we really don't care about this and this one, at least in our class. Eventually, when you do, if you want to take the MCAT, you need to understand these as well. You don't have to memorize it, but you have to understand what they mean. But we really only want to focus on the side chain, the R group. And the reason is because that's what's going to interaction between the primary structure, secondary structure, tertiary structure, and quaternary structure, which we'll talk about, I think, by the end of this week. So the interaction between each other, kind of like the hair. So the hair will interact with itself, which will create that curly or not curly. This one, it's important, but once it's in the bond, it's going to disappear. So like I said, we're going to focus on, so here's an example of these two. So even though this doesn't have OH for a disulfide bond, it's also polar, so it does have that. So this hydrogen could disappear. This one could also disappear as well. So it could be O negative, and this could be S negative. But I do want to focus on these two. So we're going to use aspartic acid kind of to show you what's going on in something called a titration curve. So here's the titration curve, and what it means is if we start off with a pH of 0 and we steadily increase the pH, we're going to see a change in the structure. And every time it changes, it kind of flattens out because technically this is called a buffer. So it's going to take a lot of more base to kind of, as we're going up, to increase the pH. Now you'll notice that there's different places, and at each of these places is where we're going to have a change. So at pK1, this is where we're going to have a change. You'll notice right here, this OH is going to be removed. It's going to lose its protons. It's going to be COO minus. So at this PA, so what does that mean? So if the pH is 1, it's going to be in this form. If the pH was greater than 2, let's say 3, it'll be in this one. So pH of 1 will be here, pH of 3 will be here. The pKa is where it's going to be balanced between that. So anywhere above will be this form, below will be this form. All right, we can keep going up. So now it's here. I already lost it. Now here at pK2, so this is around 4, you'll notice that this CO will lose it as well. So now we're talking about the R group. So here's the R group right here. and it's going to lose its proton as well and become negative. So like I said, we really don't care about these two. And the last one is going to be the amine is going to give up its protein. So it's going to be, here it's going to be 2, and here's that. But we really only care about this. So in most cases, we care about the one in the middle. So when we go back to that chart, you'll notice that this side is the small number. So this is the carboxyl group. This is the amine group. But we really only care about this. These are the ion groups. So most of these are kind of either really high or really low. This is your acid. These are your bases. And we want to know. So if the pH of our blood is 7-ish, then we should know what kind of charge these are. And those are called, and that's when we look back to these, it says physiological pH. So in our bodies, these are going to be negative. In our bodies, these are going to be positive. So when we're making amino acid groups or proteins, these bases are going to be positive, these acids are going to be negative. So those are really important because those two can now form a bond with each other and create something. All right, so hopefully that makes sense. Let me kind of get back to the last one. And here, my last slide, I wanted to show you, we really don't care about this group. This is the carboxyl group, and this is the amine group, because when they make a peptide bond, they're not going to be positive or negative anymore. Now back to this slide. Well, why is that important when it comes to pH? So you'll notice that certain proteins are designed to be a certain way, work in certain pHs. So pepsin's in our stomach, so very acidic. It works. This one's in our intestines, so you want to have something more neutral, so it's going to work best here. So these proteins are designed to have a certain shape in certain pHs. So the shape that it's in pH 2 is going to be the most effective. As pH changes, it's going to change those ionization of those R groups, so it's going to change the shape of the protein, so it's not going to work as well. Well, on trypsin, since it's designed to work well in our intestines, it's going to really be focused around closer to neutral or closer to basic, and it's going to work well here. But if it gets too acidic, it's going to also change the protein, and it's not going to work as well. So that's kind of what I want to focus on today. Tomorrow we'll kind of focus more, or next time we'll focus more on the amino acid peptide bond, which then we'll talk about primary structures, secondary structures, and so on.